There it is. All right, so I'm going to try to go through this fairly quickly because I have about I need to shorten the time so we can catch up. So I'll just get started. What I'm going to try to go through first initially is just the sort of an update of some of the new data sets that are in the gEAR. It actually turns out to be several of the ones you guys have seen and heard from today. And then cover some basics for people who have not used it yet to hopefully get you started just by creating an account, how to do basic gene expression searches, how to change profiles, what a profile is, so you can see different data with your searches, and get a high level view of just the different tools and things that are available in the gEAR. And so I'll go through these sort of as slides. I'll show you at the end, there's videos like walkthroughs of how to do all of this. So if we don't have time, of course, to go through them in here, but I'll show you how to get to them on our website. One example of what I mean about sort of the data that you see in this conference being available in the gEAR for you to interrogate. This is an example of some of the things that Dr. Kelly and Sanders were showing. This is figure 1. Here is the quote. There it is. This is figure 1 from their paper. And we see this and we hope that this is exactly the sort of thing that you can do in the gEAR, is take things that are figures in the paper and then further interrogate it yourself and hopefully visualizations that are fairly close. So in this case, we have a UMAP display. At the top, it's a nice guide so you can see which clusters or which cell types, and then individual genes and how they appear within that t-SNE as you go through. And then, of course, a heat map on the side of different marker genes. So if I sort of expand that part out, these are, again, directly from the paper. In the gEAR, we have this data set's already present, and we have created displays that are similar. So this is what we call just a side-by-side t-SNE, where you get the same sort of map to show which clusters are which. And then spatially, of course, these are opposite but the same. We can see the gene for NeuroD1 and CLB2. And of course, they correspond to what they look like in the paper figure. So that means you could then go load that data set in the gEAR yourself and keep looking at other genes you're interested in and not just the ones that are in the figure. The same thing with the heat maps. So I took just a selection of these because there's a lot of genes, but we showed them 2 different ways. So the same selection of genes in the heat map, the axes are flipped, but also violin plots instead of heat maps. You can see whichever views you prefer, both are available. And definitely given how small this is, just interrupt if you guys have a question as we go. We don't need to wait until the end. Another one is from Creighton University. Dr. Jean Zhao and David He's labs. We have this profiling mouse cochlear cell maturation. And this is using the gEAR, you can do XY scatter plots to look at differentially expressed genes. A recent data set looking at auditory neuron diversification. Again, we have side-by-side sort of t-SNE plots or UMAP plots. And then you can also configure things to be, in this case, violins. We have another data set allowing investigation of the changes in supporting cell gene expression for ATO1 and LKZF2 when they're induced towards generating outer hair cells. Again, discussed here. And these are all gEAR displays of those data sets currently present. This one's really new, profiling 40,000 cells from a cochlea at a variety of time points from young to old age. Another one from the Zhengyi Chen's lab. This one for reprogramming drug-like molecules leading to degeneration of cochlear hair cell-like cells in adult mice. So all these are currently present. You can investigate them on the gEAR and query them. And excuse me. Another one they talked about, Jaime talked about seeing his data set as a good way to look at differentially expressed cells. That particular data set happens to be highlighted in the upper left of the mouse models profile. You can use the menu to go to compare gene expressions for it and do an XY scatter of inner and outer hair cells. And 2 of the genes mentioned, so INSM1 and TBX2, it's hard to see how small this is, but they're highlighted in the XY scatter. And you can hover over any of the other genes to see what those are. So it's a very quick way to get access to that data. So like if you go to where your data set is, I'll show this in a little bit more detail, but If you click the upper menu, you can access all the analysis tools for that data set. One of them is compare gene expression. And that takes you to here. And you just pick the X condition. In this case, I did and the Y and then hit plot. And it shows you the whole scatter and it's interactive. Okay, so to back up a little bit, that's the data that is new and currently available on the gEAR, but let's just talk about the gEAR in general. We wanted it to be like one place you could go to and see a lot of data sets side by side and access a lot of tools for those data sets, sort of all in one place. Whoa. Okay, so the types of data we currently do support or show on the gEAR, they can be uploaded by you. Anybody can upload them, or our team will help you upload them if you need it. But it includes not as much anymore, of course, but microarray data, bulk RNA-seq, single-cell RNA-seq. We've incorporated spatial transcriptomics, and with the addition of EpiViz as an integrated tool in the gEAR, we can now do epigenetic data types like ATAC-seq, ChIP-seq, et cetera. Under development is proteomics sort of data. And then our team, either you can upload and curate, or our team does so and makes them all available. Yeah, so I'm trying to get forward, go faster. So this life cycle is something we're trying to really push, because I know a lot of people use the gEAR and they jump on and they search for one gene and then they jump off again. But the hope is that this cycle will start to happen more, is that you get your data from your lab or your sequencing core. You then take that and you upload that to the gEAR. When you do that, it's private by default. Nobody else can see it except for you, or somebody like me if you need help. But I'm not a biologist. I'm not stealing your stuff. Just help you get it in there. You then, after you do that, create displays for your data so you can make it look whichever kind of plot types and how you want it to look. Then you can arrange your data set alongside others if you have other private ones, or you can pull in public data sets for validation. Okay, you arrange those into a profile so you can see. You can then share that data set profile with other collaborators. There's share links where they will only be able to see it if they have the link. And you can do that at the individual data set level or whole profile level. Do lots of science things till you get done. Once you publish, then you can make that data set public so that everybody else can see it also. And then the gEAR itself can serve as the viewer that you put that link into your paper. You can get a short permalink URL, people are putting it in their manuscripts, and it becomes the sort of free data set viewer for everybody else. So that's sort of like the life cycle of data sets that we hope to encourage more use of. To benefit the most from the gEAR, you create a free account. You don't have to, you can use the gEAR without it, but just different parts of the site require the association of actions with a user account. So if you create a free account, you do so just by the upper right-hand corner, creating a username and password. It's a simple form, no credit cards or anything, of course. Just all your contact information. And then, whoa, it does jump forward, doesn't it? Okay. There we go. Once you do that, you'll see your name in the upper corner, and different parts of the site now become available that were just hidden before. From the homepage, the sort of the general layout is if you have a gene-centric view of wanting to interrogate the site, this whole area is for you. You can enter one or more gene symbols or choose a gene cart, which is just a collection of gene symbols. Choose a profile. So a profile is just a focused collection of data sets. We've got many that we curated already. So there's vestibular profiles, hair cell profiles, et cetera. And you can do a search to get to the next page. Or if you're more interested in what data sets are currently in the gEAR, I want to see that. That's how you start from over here. You can enter terms, or there's a whole data set explorer that we'll see. Tools are outlined here if you want to go straight to analysis tools. And then this whole area is all about teaching. So there's walkthrough videos of different topics that you can learn how to do it further. So one example, you just joined a lab, you want to learn about the transcription factor, PAL4F3. I want to see where it's expressed in the inner ear. So I search in that first left area for PAL4F3. This is the page that results. By default, you get this hearing profile that's probably about 12 data sets that we've curated. And you can see its expression in all of them. There's annotation display at the top. And you can switch profiles if you want to look in different data sets. There's just a drop-down menu. If you click another one, everything just automatically updates. And it's usually fairly quick. Yeah, so this is just a little bit closer view. Here's what the profile switcher looks like. If I click the top of it, there's this hierarchical thing where you can find other ones. We've added this plugin for deafness gene annotations. So if you're looking at a particular gene and we have an entry for it there where there's known deafness loci in mouse, human, or indeterminate, you just elsewhere, then if you hover over them, you get both a list of phenotypes that are known, and you can click out to external links like MGI or anything, OMIM or anything that we have external. A key thing if you're looking at any data set in the gEAR is what we just call the context menu in the upper corner. So you can click the magnifying glass to look at that one particular data set. It sort of fills the page, but otherwise this drop-down menu is how you access all the other tools for that data set, and it will jump straight to it for that data set. So displays, that's if the author of the data set has curated, or we've curated more than one way to look at it, maybe a bar plot, maybe a line plot, maybe a t-SNE. You can toggle between them all on the fly. Yeah, there's a note-taking tool where you can take notes privately or publicly on that data set. Information box gives you anything the authors have submitted, so much longer descriptions of how the data set was generated, things like that. Links out to PubMed or Geo. And then we get into the tool area. So comparing gene expression is how you perform those XY scatter plots. You can curate and make your own displays of how they look, both in single gene or multi-genes at a time. And then we have the single cell analysis workbench. And finally, we recently added, people wanted to just be able to download the data directly. So you can do that two different ways. The original bundle, whatever files they gave us, or this binary format called H5AD that's popular with Python or R. So we showed how to look for one gene, but we also added support for displaying multiple genes at a time. So instead of just one, if I search for two separated by commas or spaces, it would by default show them listed one after the other. And you'd have to click back and forth between them if you wanted to see the expression change. Instead, there's this icon here. And if you toggle between it, you can also search for families. So I can just say PAL4F and make sure exact match is off, and it will find any that begin with PAL4F or contain it. And then if I toggle multi-gene displays on, it will show all the results at the same time in the expression form. So that's when the display switch to things like heat maps and things that are appropriate for seeing multiple genes at a time. One of the recent data sets I believe was talked about here. This one was plotted with cochlear hair cell gene CART. And another view was plotted with CART for type 1 SGNs. So a closer look at the XY scatter things. Hello. From one of the data sets, you can choose compare gene expression. That takes you to the page where you choose X and Y conditions or combinations thereof. It immediately generates a scatter that you can look at. But you can also do things like do custom labels. If the data set has duplicates or replicate information, you can choose filters and significance tests. And it will either filter the dots out that don't match statistical significance, or just colorize them so you can still see all of them. And again, these are all interactive, so you can hover over any of them to see which genes they are. If on the bottom part, you can't see it just below here, but there's an area to type genes of interest. If you do that, they will appear labeled just like this in the plot. Sometimes if it's too dense, they get covered up. We're trying to work on that, but you can see all of them labeled. The plot can be zoomed and panned so you can get into areas closer. It's also interactive in that you can make selections of them. So if I was interested in this little cluster of genes, I can draw a box or lasso around them, and they will all appear in this table, showing you both the p-value and fold changes. But more importantly, you can download that as a plain text file or save it as a gene cart. If you click to save it as a gene cart, you can give it a name, and then that collection of genes can be used in many other areas of the website. So it's a good sort of pathway to go through. So if I then take that gene cart and go back to the main profile, the main page, I can search all those genes at once on the whole profile I was looking at and see it across all 4 data sets. Creating curations, that's just how we would create displays so things can look however you want them to look. Ideally, we've added enough controls for you to make it as close as possible to your figures. I click displays, in this case, this data set has 6 that have been created for it. So the user can just click any of them. And the owner has display. So if you own this data set, you can create all the ones you want available for people. And then below that, the users themselves can create their own. Everybody else doesn't see those, but you do, and you can have them. There's a lot of configuration options and display types, including color ranges and color scales, or in some of them, you can even do the exact colors you want things to be. You can also do multi-gene displays. So it sort of reads into the data set and detects what sort of plots are supported. But there's several. Okay. Hopefully I'm staying on time. I know we don't have a lot. Data set collections, just like we have gene carts, which are gene collections, to do data set collections, you want to find out what data sets are already there and be able to build them into a collection and organize them however you want so you can see them side by side. This thing called the Data Set Explorer allows you to do that. By default, it opens up and shows you most recent public data sets opened. But what should be sort of the familiar UI, you can do faceted searching on the sides to say, show me all public data sets that are zebrafish that are also single cell RNA-seq. So if you just click on those, it'll rapidly filter the list to just those. You can use the profile controls to build your own profile. So as you find data sets you want, you can just use the plus signs to add them to it. If you look at any one of the entries in here, what buttons are available depend on whether you own the data set or not. So if you own it, you can do things like deleting it, editing it, but otherwise you can view a data set directly on the homepage, add or remove it from the current profile. There's a sharing button. So that's what you use if you want to send that directly to somebody else. Or you can access all the analysis tools from here also. This is similar to the upper right corner on the front page. There's an expansion button on the list that comes back. If you click it, it opens up even more information, which could be a very long description. We don't really limit it. I think it's 65,000 characters or something you can put in there if you wanted to. But it also shows if that profile exists, if that data set exists in other profiles, it shows you the list of all the profiles it's found in. So you can click on one of the profiles and go straight to it. You can share the whole profile instead of just one data set. This is probably the key one for if you want, if you build a profile that has your data set in it and others, you can then click that link and that's what you could share with a manuscript for a submission. Okay, and then going on to the workbench, you see you want to generate clusters, things like marker genes. If you choose a data set that somebody else has uploaded, like one of the ones we've heard of today, it automatically loads whenever you load a data set. You can start from the beginning and create your own analysis, but first you can use the drop-down menu and check, does the author already submit one? So when we download the data sets, if they already have clustering data or TSNE or UMAP, we maintain that exactly. We don't reprocess it like NCBI does with the genome. We just take their exact thing. And it's available under this primary analysis tab. So if you choose that, it skips all the steps up to that point, and you can go straight to doing things like finding marker genes and comparing clusters. So if I choose it to show me the top 10 marker genes in each one, you get that automatically. You can then click all the ones you're interested in or type in additional ones and visualize to visualize those markers. And this is pretty quick. Almost all this from loading the data set to doing this step is usually just 2 or 3 minutes or less. If you want instead to do your own full analysis from the beginning, such as if this was your data set you just uploaded, you can do that. The menu on the left shows you the steps. So you click, Sorry. There you go. All right, if you click a step on, a form will appear and you just answer the questions in it and hit submit and it computes on the fly. And so on as you go down each step. So each one has graphical results as you go down each one. So principal component analysis, and there's information to guide you for what each of the options mean. You can do TSNE or UMAP or both. Then clustering options. You could name the clusters if you the combination of the clustering and showing you the marker genes in each cluster. A lot of you know what that means, which ones are. So then you can name, instead of it being 1, 2, or 3, you can give it proper names. And that's what everybody else will see. Yeah. Okay, and then trying to go through. One of the more new tools is transfer learning. This is an R package, a collaboration with another group at Hopkins called Project R. It sounds like it may be something similar to the Symphony package described in the last talk. But what it means is you can take patterns that are learned and use that with a new data set from a target data set. So you can do things like principal components learned in one and use that as molecular signatures for the next data set. Really good if you're doing several many in a row. In this example, for example, the principal components and the clusterings are then mapped to the next data set that comes in. So it's really useful. It's a really quick way if you have sort of like a lot of experiments to go back to map the work that you've done on one data set onto the next one. Uploading can be a little bit tricky. This is something that's being completely rewritten. It's pretty much consists of 2 steps. There's just a plain Excel file that you add metadata to. That's usually the easiest part. And then the second one is expression files. We take most of the common formats, MEX formats, 3 tabs, things that come off the machines directly. And it validates it as it goes, which can be a little bit slow sometimes, but it validates as it goes, and then your data set is just ready. And of course, we're always happy to help with that. And one of the newest things is we've added EpiViz integration. So one of the data set blocks and profile can be an epigenetic viewer. You can do things like, it looks kind of like a standard genome browser where you can lay upon the genome ATAC signals, ChIP-seq signals, et cetera. And that's just alongside all the other data sets that are there. My goodness. There we go. Just a reminder, I know we had to go through a little bit fast, but there's many different ways to get through the tutorials. There's a user guide at the top. There's a video right smack in the middle of the homepage for where the introductions go. And then topic-based ones are all listed on the side. And on many of these, we have not only videos, but also if you prefer slides or slides that guide you through the same thing as a video. Some future work that we're working on now. One of them is a lot of people have requested sort of robust support for groups. So like your lab group could create an organization within the gEAR. You could add users to your lab group. And then if any one of you upload something within your group, everybody else in your group can see it. So it's private still, except for within your group. Another one is better highlighting what's new in the gEAR. So this may be released in the next just couple of days. I've already done it, but this block on the left is becoming something similar to this, where it automatically shows the most recent public uploaded data sets. So you can just go to the gEAR and see new ones that you guys hopefully have uploaded. And then a much bigger effort, and this one makes me excited. It's at the University of Maryland College Park. They have a human-computer interaction lab. And for master's students of essentially UI design, they created a gEAR capstone class where these students, their whole project was the gEAR for a year and doing a UI redesign, talking to users, lots of sort of going back and forth about things. It's been a fun process to watch, but creating all new looks for every page of the gEAR that hopefully make it much more user-friendly. And this is an example of what one of them looks like, sort of a landing page where the menus on the side are always consistent. It's actually themable. So we have a color where it's all white, one where it's the standard purple, another one where it's like a dark theme. You can access tutorials from the top, much more advanced search place at the very beginning, and things like we track what you've done recently, so you can jump right back into recent work that you've done. This is another page. If you search for a gene, something like what it must look like. The left menu collapses to only be icons, but you can expand it whenever you want. And a different view of what the scatter tool will look like. And again, it's all themable, so you can make it look like how you want it to look. But it's been a fun project to do, and during this year, we'll be actually implementing the results of it. So the gEAR team, myself, another engineer, Sean, is on here. I can't see. There we go. There's usually just two engineers on this project at any time. Dr. Arzano, and our curators, Yang and Joe, and then everybody in Rona's lab and collaborators that have helped make the tool that it is. I think I did that faster than I was supposed to. But yeah.
Yeah, so after you analyze
Yeah, so you mean somebody else, like somebody else has uploaded the data and then you do your own analysis? I mean, it's public data at that point, like any others, so you should cite them for sure. But other than that, yeah, it's public data at that point. And if you do an analysis yourself, like with the single cell workbench, you can also share that analysis. So like if you do a share link to that, when somebody goes to the page, it will preload automatically with all the graphics that you saw, and they can see what you've done and interact with it. Yeah.
You think you could I don't know how to put that in there. You guys have, I know you have for a toxic, yeah.
Yeah, so just sort of a more expansive integrated genome viewer in association with, because EpiViz is kind of doing genome views, right? But it's limited to mouse. You're one looking for it for anything.
So I'm just trying to see what public knowledge, how to compare it, but anyone who's going to displays variants, maybe we use it.
Okay. Yeah, I mean, it's a good idea. Right now we have things like links out where it would show that gene like via the annotation section. There's many links out available that would link out to places like NCBI and stuff who do have that visualization, but I don't think it's unique plots. It's more like genome browser type displays where they would see splice sites and everything else, but that's it.
Finding out what's more strongly spread in the cell line or whatever.
What's the most strongly expressed cell type? Yeah, that's typically the like clustering and then like UMAP and T-SNE displays, right? Okay, yeah, we can meet after this. I can show you. Yeah. I have my huge screen laptop, so I can I can walk through. Yeah. We don't currently. There's like a microRNA tree browser as part of the annotation, but that's it. That's the only thing so far. For the ones that are related to it. Okay. Well, thank you all for staying so late. Yeah, that's a good question. So some have already started. The tricky part is how to do it in a way that everybody will do it the same way, because right now people are, we sort of accept whatever data people give us, and we don't kick out any genes or any rows or anything like that. So some people are putting expression data with alternative splicing, and they're just calling the gene symbol like -1, -2, -3, something like that. And we take it and display it. It works, but there's no like consistent way that people are doing it uniformly that like we're really like showing that it's an alternative splice. So it's all there and it's all present, but it's not really handled in a clean way yet. Yeah, so it's definitely an important thing. Okay, well, thank you all. I think got a few minutes before the